Are you struggling with cell communication and cell cycle? Then you're in the right place. Because for the next hour, we're going to be talking about all the parts of cell communication. We'll talk about mitosis, we'll be talking about meiosis, and we're going to just get it so that you are more prepared and you are ready for that AP exam that's happening on May 11th. So without further ado, let's get started. So first thing, who am I? Why am I even here? So my name is Tiffany Jones, and I'm from AP Bio Penguins. And so I'm here just to help you to understand cell communication. And then also I'm here to help you to talk about mitosis and meiosis. So I run a free AP review account on Instagram, and I do some stuff on TikTok. I do it all over the place. So why are y'all penguins? Like, why do I even call y'all penguins? So penguins are because they're in their little tuxedos and they're dressed for success. So I feel that students are also dressed for success. So I want to make sure that you know that as an AP Bio Penguin, you're dressed for success. So welcome to being AP Bio Penguin now. So today we're going to go through a couple different things. We're going to talk about cell communication. We'll talk about the cell cycle, specifically looking at the steps of the cell cycle, as well as those checkpoints. We'll look at mitosis. We'll look at meiosis. And then we'll do some practice questions. And then we'll have the Q&A at the end. Now, I find that there's a slight delay between what I say to you and what, like when you finally see it. So there's someone that's going to be in the chat to try to help you out. So Miss McClinton is in our chat. She's our chat Q&A. So if you have a question as we're working through this, just be sure to ask those questions in the chat, and she's going to help you out to make sure that everyone is all on the same page. But then during that Q&A, if there's anything that didn't get answered, we can talk about those at that moment. So are y'all ready to get going? I'm excited. So cell communication, there's three main parts to cell communication. So we have reception. And during the reception, this is when you're going to have your ligand, your signaling molecule. This thing is going to bind to a receptor. That receptor can either be sitting on the membrane or the receptor can be on the inside of the cell. It all depends on a couple different components. Once that signal binds to our receptor, which is a protein, it's going to have a conformational shape change. It just changes shape. As we looked at before with proteins, if you bind to a protein, the protein automatically changes shape. That's that induced fit model, right? Or your teacher may have called it the locking key. But the big thing that you say here is conformational shape change. So the example that the CED uses is the G protein coupled receptor, which is going to be found in the insulin signal transduction. So we'll see the insulin molecule will bind to that G protein coupled receptor that then will turn on the G protein using GPP, which activates this huge phosphorylation cascade to then release more glucose. So it's this whole big thing. So there's two types of ligands you can have. You can have a steroid hormone. Steroid hormones are going to be, as we know, steroids, and they're going to be made up of four fused rings that are nonpolar. So because of the fact that steroids are nonpolar, we're going to see that when they get released from a cell, they go through simple diffusion. We're also going to see that when they reach their cell, the cell that they're supposed to, like their target cell, the receptor for it is going to be intracellular. It'll either be in the cytosol or it's going to be inside the nucleus. And the reason why is because that steroid passes directly through the plasma membrane. So the receptor for it has to be on the inside of the cell. So examples of these types of hormones are going to be testosterone or estrogen. There's actually an FRQ question from 2017, I believe, in which they looked at estrogen and we're looking at antibodies and we're trying to inhibit this estrogen molecule. So we talk about that the antibodies can't reach that receptor on the inside of the cell because the antibodies are polar, and then the receptors on the inside. So there's a whole big thing with that if you want to look back at that FRQ. The other type of hormone we can have is a protein hormone. So protein hormones are going to be, of course, polar. So in order for them to be released from a cell, they go through exocytosis, and the receptor for them are going to be extracellular. So these are going to be receptors that are on the membrane. So now the students have trouble with this. They think that the signaling molecule moves throughout this whole process, and that's not what's happening. The signal molecule binds the receptor, and that's the end of the ligand. So after that, it's all about a domino effect of one thing that does something to the next one, that does something to the next one, that does something to the next one. So the actual ligand doesn't leave the receptor. It kind of stays the receptor and then pops off to turn off the signal. So an example of a protein hormone would be, of course, insulin. So now that we've received the signal, we've got that message. Now we go through transduction, which is where we see this amplification of the message. We bring it from one location to the next. The big part is just to amplify that signal and transduce that signal, get it from receptor to the response. So this can happen through a series of different things. One thing you can do is a phosphorylation cascade. And in the name, it tells you what it does, right? We're going to see phosphorylation taking place. So one relay molecule will get phosphorylated by another one and so on and so forth until we finally bring the message to our response. So with a phosphorylation cascade, we're going to use something called a protein kinase. That word kinase is important. Kinase is an enzyme, and that enzyme is going to phosphorylate something. And we see that like in many different places. There is a receptor called tyrosine kinase, which means that we're adding phosphate to a tyrosine, basically the amino acid on that receptor. So you'll see this word kinase a lot. Kinase just means we're phosphorylating something. And so since this is a protein kinase, this means that I'm phosphorylating proteins, okay? And so you'll see like this long list of just things getting phosphorylated. And I have an example to show you in just a little bit that shows that phosphorylation. Other ways that we can go through transduction is where we look at secondary messengers. Okay, so if there's a secondary messenger, that means that there's a small little molecule that's being released that's going to travel throughout the cytosol to go to cause some other type of response. So the examples we see are calcium and cyclic AMP. Calcium we see in our muscle cells. So if you remember back to unit 2, we talked about the smoothie R and that the smoothie R would hold calcium. And so from the pseudotransduction, from the transduction pathway, we're going to cause the smoothie R or sarcoplasmic reticulum of our muscle cells to release that calcium. That calcium will then move to bind to a troponin complex on your muscle to then cause the contraction, right? And so if we see that contraction taking place, right, that's now our response, right? The muscle contracting. Another example we have is cyclic AMP. Cyclic AMP we already looked at with the insulin pathway. So once we move down the pathway of insulin, we'll release the cyclic AMP that then activates another protein kinase to continue this pathway to cause us to release, of course, the glucose. So that just gives you an example of our secondary messengers. And the last thing we have is response. So with response, there's a couple things that can happen, but you're going to have to be given something. So on the AP exam, they're going to give you a pathway. And then you have to look at that pathway and be able to determine what that response is, determine what happens if a intermediate is mutated, determine if you have an inhibitor, what happens. So you need to just look at the diagrams. And so we have 2 examples I'm going to show you in just a moment that really helps us to see how the College Board can give you the pathway or whatnot. So you're not going to need to memorize any pathway. You just have to have a general understanding of what's taking place if you look at the picture of it. Okay, so different responses that are directly out of the CED, it can cause cell growth, so the cell can grow. It can cause secretion of molecules, so causes it to go through exocytosis, causes it to release, like we look at neurotransmitters, that's how they release, it's from signaling molecules. Talk about gene expression, so it can be a transcription factor to turn on or turn off the transcription of a certain gene to make a certain protein or not make a protein. And then of course, apoptosis, which is cell suicide or cell death, right? Programmed cell death, where we see that the cell is going to break down from the inside out. And so these are just some examples of things that you could see on that AP exam. So this was on the 2013 exam, and they just asked you to describe the pathway. So first step was, of course, reception. In reception, we see that we have a protein hormone, this ligand, is binding to a membrane protein due to reception. That binding then causes a conformational shape change that initiates this transduction. So nothing new at that point. But you're trying to come up with, what do I see in this picture, right? Step 2 is that transduction. So the point of this, we're just amplifying that message. We're transducing that message from reception to response. And in this process, we can amplify that signal, and we can kind of ensure that we have any type of interaction that needs to take place to ensure that I go through the multiple pathways. And then the last thing is response. So based on this picture, I can see that I went into the nucleus and that it's acting as a transcription factor. So it's going to either turn on or turn off a gene. So to initiate or inhibit transcription. So that's just all we're doing is looking at a picture and being able to say what's happening. So there's a lot of different things that they can do with this. Here's another example. So this is off of the 2021 exam. It's a free response question that they had. There was a whole backstory looking at kidney molecules and looking at materials being moved across membranes. And so I wanted to just hone in on the actual like signal transduction that we see for this example, right? So we see that we have this molecule, this ligand, it's going to be bound to this sodium potassium ATPase. And then we see these little arrows. That's just showing us the transduction. I see this MEK that's being phosphorylated to become MEK. And then I have an ERK that's also being phosphorylated to make PERK, okay? Or ERK to PERK. So what you see here is this is a phosphorylation cascade. So this got phosphorylated, which then causes the next thing to get phosphorylated. That's the big thing you're taking from this diagram. So the question is based on this picture, predict the change in the ratio of ERK to PERK. So the ratio of this molecule to this molecule, if we have an inhibitor that is going to inhibit the PMEK, so inhibit this P, whatever this word is, okay, from being able to phosphorylate this process. So the inhibitor isn't going to take place here, right? So I'm going to have a higher amount of ERK to PERK when the inhibitor is present than if it was absent, right? Because if it was absent, then this is going to kind of add as an enzyme to this process and make more of that PERK. So what they wanted you to be able to understand is if you don't have this enzyme right here to speed up this part of the process, you're going to see a backup of the ERK. There's not going to be as much PERK because of the fact that enzyme that speeds up that pathway isn't there. So you would find that there is going to be an increase of the PERK to the ERK, there's an increase of the ERK to the PERK. Or you could have just said that it stayed the same. A prediction point is just asking you to predict something, right? So when you're given a question that asks you to predict either increase, decrease, stay the same, you need to at least give something. Sometimes 2 of the 3 options can be given in the prompt, in the answer choices. That's something you could have put. So always try on these predictions. The second part of this was a little harder, right? You had to justify, like, why was that? Well, as we said before, if I don't have this enzyme to speed up the pathway, then this ERK is not being converted into PERK, so there's a higher concentration of ERK in the cell. So I know that sounds a little weird when I'm going back and forth between ERK and PERK, but I hope that I slow down enough that you can kind of follow that logic. The second thing we have as an example is the 2018. Now remember, if you have questions, you can pop them in the chat. Ms. McClinton is there to answer any questions you have as we go through this stuff. So the 2018 question, it was about our response to an infection. Now the immune system is no longer on the AP exam. So like as a official standard in the within the CED, it's not actually there anymore, but it can be used as an illustrative example. So they can give you a question like the 2018 question that doesn't actually require you to know anything about the immune system, and you can still be able to answer it. The only part I think of this question you couldn't answer was maybe part D, because it asks a specific thing about the immune system. But other than that, the rest of the thing is fair game. So I wanted to show you this question to kind of show you how the application of a signal transduction could be, right? And so they say that in response to intracellular pathogens, the inactive caspase 1 is cleaved and forms this caspase 1, right? And so that's going to then cause this, active interleukin to be released, as well as it causes the gastrodermin to become enteromal gastrodermin that then makes this pore in the membrane, right? So a lot of that was given in the passage. It could just be a diagram, or it could be written in a passage. This question gave it to you in both facets, so you can actually see it both ways. So they said, well, describe the effects of inhibiting step 3. So this step right here did not take place. There is not this step taking place right here. Like, we inhibited it, we stopped it, we slowed this process down, no go, right? What would happen to the formation of the pores? Well, if this pathway doesn't take place, can you make a pore? Well, no, pores are not going to be formed because I inhibited this step in this pathway. I inhibited this transduction step, right? I can't do that response of forming pores because I couldn't do this step. So then they said, well, what happens to the interleukin release? This has no connection to this pathway. So remember I said that sometimes in transduction, you're going to have multiple pathways. This is an example of that multiple pathway thing, where maybe one step gets inhibited, but the other step is not. So because of the fact that this step right here is not inhibited, we're still going to see interleukin getting released. It's not affected. You'll have the same amount released and will still cause that immune response that we would have seen otherwise. Now, there is a question in the chat. It says, in what way could the ratio of ERK to PERK stay the same? On that previous question. So the PERK to ERK could potentially stay the same in the aspect of that enzymes are just speeding up the chemical reaction. So you could have just said that, well, it's going to have no effect. It'll stay the same because the reaction will take place at the same rate, no matter if the enzyme is there or not. Well, not the same rate, but it can still take place whether it's there or not. So depending on the time period, we could still end up with the same ratio. There was no time period given in the question. So it doesn't actually have that constraint on it. Okay, so we've got that done with signal transduction. On to the cell cycle, yay! So the cell cycle, this is a general gist of the cell cycle. It's just the life of one cell, like how long it goes from when it was made to when it divides again. So there's technically 3 different parts to the cell cycle. The first part is interphase. Interphase is made of 3 parts, G1, S, and G2. The second part we have is the mitotic phase, or kind of you could think about meiosis, okay? So we just say the M phase. And then after the M phase, we have cytokinesis. The thing the students need to make sure they understand is that when you ask about mitosis or you ask about meiosis, interphase is before it, cytokinesis is after it. Mitosis and meiosis is literally only looking at that nuclear division. It's only looking at that nucleus dividing, okay? Versus interphase is getting prepared for that. And cytokinesis is the division of the cytoplasm. So that's how you get multiple cells being made. But the actual M phase or mitosis meiosis is literally just the nucleus, okay? So if we look at this, G1, S, and G2, all through interphase, the cell is going to grow. The cell grows because at the end of this, we're going to basically pinch the cell into 2, and we want that cell to be approximately the same size. So the cell is going to actually grow as we go through G1, S, and G2. Some students just kind of remember the G1 and the G2 as grow. They're actually gap 1 and gap 2, but it all depends on how you want to kind of remember it to kind of keep yourself straight. But it goes through all 3 stages. So in G1, this is when we duplicate our cell organelles. So we'll kind of make copies of the organelles, like the mitochondria and the chloroplast can replicate, of course, by binary fission, while the other ones can get formed by the cell. So as we know, the ER is all made from the extensions of plasma membrane. We know that the lysosome is just enzymes being formed that come out from the Golgi. So that's how we form our organelles during G1. We also synthesize different proteins, we synthesize RNA, and then the different building blocks for our future cells, okay? Yes, super important. This is when we're going to go through DNA replication. This is when we have the 1 copy of DNA, we make a whole other copy of it. So when I divide my nucleus, I will have 2 sets, so they can 1 go to each cell. And then G2 is going to be continuing this synthesis of our proteins and our RNA. It makes those organelles, and then it just kind of reorganizes the components so that we're kind of ready for that division to take place. Mitosis, or meiosis, depending on which one you're looking at. Well, right now we're just looking at mitosis. Has 4 main steps. You have prophase, metaphase, anaphase, and telophase, okay? Now, I have a cell cycle dance because, of course, I have to make all my things in my life interesting, right? So we start out with prophase. Prophase is when the cell is preparing to divide, okay? So we're going to see our nuclear envelope is going to kind of disintegrate. So you see this kind of like a dotted line that's showing the nuclear envelope is kind of disintegrating, breaking apart. Our sister chromatids are going to everything's going to condense. So our chromatin, which was our DNA and our histones, the DNA wraps around those histones, it gets super condensed. And then the sister chromatids find each other. So this right here is showing you basically those sister chromatids that's attached at its centromere. This cell has actually 2 total chromosomes, so 2. And so you see 4 because there's the single chromosome plus its duplicate, which was its sister chromatid. So that's prophase. Then we move into metaphase. This is when our sister chromatids are aligned on the metaphase plate. They're lined up basically in the middle of the cell. The way they got there is because we had these kinetochore microtubules. The microtubules come out from the centrosome. They bind to the kinetochore in the centromere of the chromosome, and it attaches to it. Okay, so at the end of this step, we kind of have this little tug and war that takes place. Due to the tug of war, you get the sister chromatids lined up in that middle. Then we move into anaphase, which we're going to move the sister chromatids apart to opposite poles. So you see that our microtubules are going to go through depolymerization. They get shorter. And as they get shorter, it then kind of reels the sister chromatids to opposite poles. And then telophase is when I make my two new nuclear envelopes, right? So we now have our two nuclei surrounding our two things, right? Now notice that we still only have the one cell because I haven't gone through cytokinesis to divide my cytoplasm. So just to make sure we went through that dance correctly, prophase, metaphase, anaphase, telophase. And then of course I have a fun thing to do with cytokinesis, but we're gonna hold off on that one. And then cytokinesis is when we're going to split our cell into two. Basically, we're splitting the cytoplasm. So for our plant cell, that involves making a new cell wall by distributing, basically the vesicle drop off this little cell plate and then it forms a wall. And then when we look at the animal cell, it goes through a cleavage furrow. There's actin that's going to basically squeeze, which pinches it into two, which then, of course, divides our cytoplasm. So if you were to think about the whole cell cycle dance, we have G1, S, G2, then we go through prophase, metaphase, anaphase, telophase, cytokinesis, then we just kind of divide the cytoplasm. Lots of fun. Kinetochores also move along the microtubules because the kinetochores are part of the centromere. So like basically it's a protein that sits at the centromere of our sister chromatids. And so the microtubule attaches to that and it kind of pulls it. The best way I've explained it to my students is it's like you have a belt around your waist and then you hook, basically take a hook and you hook up to the belt and that's what pulls you. So it kind of pulls the two apart and splits them. So that was basically the overall cell cycle. Now, we have to regulate our cell cycle. If regulation doesn't take place, that's what leads us into cancer. So we have a couple checkpoints to ensure that we get through all the parts of the cell cycle appropriately. So G1, this is going to be our go-ahead signal. We get a signal that says, hey, it's okay for you to divide. Hey, all the DNA is good. There's no damage. We have adequate resources. It's usually a growth factor that takes place. If it doesn't get the go-ahead signal at the G1 checkpoint, that's going to cause it to go into G0. It's a non-dividing state. So there's a couple of your cells that are in this, like your nervous cells, they're in a G0. They're not dividing. There's other cells, like your muscle cells aren't dividing. But there's other cells like your liver cell that's currently sitting in a G0. It's not going through a dividing state, but if given a signal, it can go through a division. So that's how you can donate part of your liver and it will regenerate. So here we kind of see that, right? So we've got cyclin that's going to be taking place through basically this whole process. Cyclin is just a signaling molecule. It's going to bind to a CDK. CDK is cyclin dependent kinase. Ooh, there's that kinase word again. Its job is to phosphorylate. So when the cyclin is present, the cyclin is going to bind to the CDK that activates a phosphorylation of these different targets that then, of course, allow it to proceed into the S phase of interphase. And so this is just kind of a little behind the scenes what's taking place. But you need to just understand that the G1 checkpoint, you don't bypass it, you go to non-dividing state. The S checkpoint is ensuring that all of our DNA has been replicated and that replicated DNA is not damaged. So if you have damaged DNA, you don't want the cell to divide with that damaged DNA. And if you can potentially fix the DNA, you of course want to hold up, wait a minute, put a little bio in it. And you want to kind of like fix the DNA before you get going, right? So it detects problems in the DNA, cell cycle will be halted to complete that process. Or if I need to finish going through replication, it'll halt it. So P53 works a whole lot in this. And so they call that like the guardian gene because it's going to ensure that there's no like issues, right? So if there's damage in our DNA, the P53 is then going to make this CDK inhibitor. So the inhibitor basically stops this process from taking place. So we're not going to go into mitosis because I'm not ready to divide yet. So you're also going to see that the cyclin level is going to increase to a certain level while CDK stays equal. So I have seen some questions on the exam where they ask you, where are you in the cell cycle based on that cyclin level. So if you see the cyclin level increasing, it means you're in that interphase and it's going to increase until it reaches the high, like a certain concentration. And that's what then initiates you to go into mitosis, or it's actually called MPF, or maturation promoting factor. So now we're into mitosis, we're ready to go. And we mentioned about the kinetochores and the microtubules, and we said that they had to be attached. Well, if they're not all attached, that's a problem, because when they pull apart, the microtubules pull apart, you could end up with an unequal division of your DNA, which is a problem. So in order to kind of protect it, what we do is that we are going to check that all of our sister chromatids are attached to spindle microtubules. We have to make sure that every single one of them is attached. If they're not attached, that's going to cause basically the hold up signal, right? So you're going to see that there's this pathway that's going to kind of basically stop and wait sure it's ready. When it's ready, it's then going to break the cohesion that holds our two sister chromatids together. Once that has been broken apart, then we're going to use separase, which is going to be an enzyme that separates them. And then voila, they're now going to move into opposite sides of the pole, okay? So again, the big idea is to remember here, G1 is the go-ahead signal. G2, make sure our DNA is correct. And then our M is just to make sure that the microtubules are attached to all of our sister chromatids before we divide. So that kind of brings us to meiosis, right? So meiosis has the same thing. You have prophase, metaphase, anaphase, telophase. Problem is, now we go through it twice, right? Because I need to have half of the amount of genetic material. I don't want to have identical chromosomes. I want them to be slightly different. So we're going to notice as we go through meiosis that there's going to be special things that happen in meiosis that don't happen in mitosis that allow our cells to be genetically distinct. They're different. The first thing, prophase 1, chromatin condenses, our sister chromatids pair up, our homologous chromosomes pair up. When we see homologous chromosomes pairing up, what that has to do with is that we have the maternal chromosome and the paternal chromosome one. So these are the chromosomes that are the same length and they have the same banding pattern. What that means is they have the same genes on them. They may be different alleles, but it's the same genes, and they line up pair by pair. And so during prophase, while they're lined up making that tetrad, sometimes the non-sister chromatids will overlap and they'll exchange genetic information. So now we have a little bit of differences between all 4 of our chromatids because this one is all paternal. This one's recombinant of mostly paternal. This one's recombinant mostly maternal. And then this one's all maternal. And so it kind of helps you to see kind of we're crossing over, right? So this leads to one of those genetic distinct parts. So move into metaphase. Metaphase is going to be when our homologous chromosomes are going to align on the metaphase plate. Now, notice I put homologous chromosomes in like all caps because that's a big important thing to make sure that you know and you understand is that as we go through meiosis 1, it's always the homologous chromosomes that we're looking at. So our homologous chromosomes will line up on the metaphase plate. This is what leads us into independent assortment. Independent assortment is when your chromatids will, your homologous chromosomes will independently line up on that metaphase plate. So you can either have the maternal facing one pole and the paternal facing the other, or it can be flipped. So there's a 50% chance for each one of your chromosome pairs. So there's 23 different chromosomes we're looking at. So there's, I think, like 7.2 trillion combinations, 8.4, there's a number and I can't remember right now, but it's like 8.4 billion combinations. Whatever. So it's basically 2 to the 23rd if you're wondering what the math is. Anyway, so this is where it leaves us in independent assortment so that we can kind of then create genetically distinct cells at the end of that. Okay. So anaphase, the homologous chromosomes will move to opposite poles. Okay. Because again, we're looking at homologous chromosomes. And then telophase, I've made my 2 nuclei that have the separated chromosomes. Here's where students often mess up. You don't realize, since we're looking at basically a chromosome and its pair, you think that we're still diploid. We're not. These cells are haploid. They're haploid cells. You have one chromosome and you have basically a duplicate of that chromosome. There's no extra information. So we are haploid. We have one set of chromosomes. We have 23 chromosomes, and then we have a duplicate of it. Okay, so we're already haploid at the end of telophase 1. Then the cell, of course, the cytoplasm divides, and now we move into prophase 2. Okay, now, as we said, homologous chromosomes have split from each other. So as we go through meiosis 2, now we're looking at sister chromatids. So their sister chromatids will condense, the chromatin condenses, sister chromatids will line. Metaphase, sister chromatids will line up on the metaphase plate. Anaphase, sister chromatids will be pulled apart to opposite poles. And then telophase, the nuclear envelope forms around our new 2 nuclei. Okay. Now I went through that last part kind of fast, right? Because meiosis 2 is basically identical to mitosis. Okay, because we're looking at sister chromatids in mitosis, and we're looking at sister chromatids in meiosis. So we're looking at basically identical. Okay. But I did want to make sure that I denote for you that meiosis is looking at homologous chromosomes. Meiosis 2 is looking at sister chromosomes. Whew, that was a lot. So there have been some FRQ questions looking at mitosis versus meiosis. I did want to take a moment to let you kind of compare the two. So in mitosis meiosis, when we think about the parent cell ploidy, so both of the parent cells will start out being diploid. They're 2N, okay? And I'm only using this as the example because it'll allow us to get to the end. If your parent ends up being a tetraploid, then you would have to kind of align this information to make it match up for your example for whatever question you see. So the rounds of DNA replication. Both mitosis and meiosis will go through one round of replication. They go through it during interphase before mitosis and meiosis takes place. We'll go through that S phase of interphase and we replicate the DNA. Okay. The reason why you don't have to do 2 rounds of replication is because when you divide at the end of meiosis 1, you already have a duplicate. It's already a chromatid plus its pair, right? Plus its sister chromatid. So they're already identical and there's no reason to go through division because I already have a copy. How many rounds of division do I go through? Well, mitosis goes through 1 round of division. So it only divides once. And in that division once, you end up making, of course, those identical cells that are also diploids. Versus meiosis goes through 2 rounds of division so that I can end up with 4 cells that are haploid. Okay, so the ploidy of the daughter cells. So mitosis gives you identical cells, so they're going to also be diploid. Versus meiosis, their cells are going to be haploid. And then I have 2 cells in mitosis versus 4 cells in meiosis. The reason I have 2 cells in mitosis is because I only divided once. The reason I have 4 cells in meiosis is that I divided twice. Okay, think about it. If you have 1 cell and you divide it once, you end up with 2, right? And if you divide again, which I can't split my hands, but I hope you understand, you would end up with 2 cells from that, so it gives you 4. Okay. When you kind of compare the DNA for the daughter cell to the parent cell, in mitosis, they're identical. The point of mitosis was asexual reproduction. I was trying to make clones of those cells. Versus in meiosis, they're genetically distinct. They're completely different from those parents because now we've had crossing over take place and we've had independent assortment take place. This is one of the ways that we can allow for the genetic diversity in the population, is because of these things that occurred during meiosis. Crossing over does not occur in mitosis, but it does occur in meiosis, and it takes place during prophase 1. And then independent assortment does not take place in mitosis, but it does take place in meiosis, specifically during metaphase 1. So that kind of gives us a little bit of differences between the two. I don't really see any questions happening in the chat. So y'all, I hope we're doing okay. So let's talk about some practice questions, right? So insulin is a protein hormone that is going to be secreted in response to elevated blood glucose levels. When insulin binds to its receptors on the liver cells, so that immediately tells us it's protein, it's polar, the receptors on the membrane. The activated receptors stimulate a phosphorylation cascade, okay, so that's my transduction, that caused the translocation of glucose transporters to the plasma membrane. That's my response. So the question prompt has given me my reception, my transduction, and my response. So based on the information, which is all describes the role of insulin in this liver cell signaling pathway, right? So I see that the insulin binds to receptor. So does that mean it is the receptor? No, because it's given in the question that it binds to the receptor. It acts as a secondary messenger. No, because that's part of transduction. It talks to us that we have a phosphorylation cascade. We've already passed the insulin. Protein kinase, also part of transduction. So we've already passed that. So the only logical answer we have here is that it acts as a ligand. It's going to be the signaling molecule that binds to that receptor. So hope we're good. So second question here is about feedback mechanisms. Feedback mechanisms was in the CED for this unit, but it didn't it didn't seem like enough to make a whole PowerPoint for it. So I was like, I'm just going to put it within these notes. So the big thing about negative versus positive feedback, negative feedback is going to bring you back to homeostasis. If something gets too high, it helps to bring you back down. If something gets too low, it helps to bring you back to normal, right? So the whole big thing about negative feedback is to maintain homeostasis. Positive feedback is going to help to kind of build up to some process, right? So the product is going to come back and keep amplifying and more or less hyping up and building to that feedback, that response, okay? So the endocrine system incorporates feedback mechanisms that maintain homeostasis, during which of the following demonstrates negative feedback by the endocrine system. Now remember, endocrine system is no longer in the curriculum, but it can be used as an illustrative example. This is one of those examples. This came, I believe, out of the CED. This is an example of something that they could give you that you don't need to know about the endocrine system because everything's going to be given to you in the question. So you just have to apply your biology knowledge to it, which we said negative feedback is where the product comes back and inhibits, right? The product comes back and is going to maintain homeostasis, okay? So during labor, the fetus exerts pressure on the uterine wall, inducing production of oxytocin that stimulates uterine wall contraction. So I have pressure, that pressure causes some of your release that then causes more contraction. The contractions cause the fetus to further push on the wall, so it pushes harder, increasing the production of oxytocin. So this is an example of positive feedback, right? The response is oxytocin, and the oxytocin causes more force, which causes more oxytocin. So it's kind of building up. So that would be an example of positive feedback. After a meal, blood glucose level becomes elevated. So our glucose is too high, stimulating beta cells to release insulin into the blood. Excess glucose then converts to glycogen, reducing blood glucose level. So by me releasing insulin, my high level of glucose has now been brought back down to normal. That's negative feedback. At a high elevation, atmospheric oxygen is more scarce. In response, signals that oxygen is low, the brain decreases individuals' rate of respiration. You have a low amount of oxygen, but you're not going to respire as fast? That doesn't make any sense. Transcription factor binds to regulatory region of a gene, blocking the binding of another transcription factor required for expression. The point of transcription factors are to help with transcription. So why would I be inhibiting transcription from taking place? That's not going to work either. So our only logical answer we have here is B, because it's talking about bringing us back to homeostasis, bringing us back to normal. So into mitosis, meiosis, right? Humans have a diploid number. The diploid number is 2N, so 46. Which of the following statements best predicts the consequence if meiosis did not occur during gametogenesis? Do you know what gametogenesis is? Because I don't. I'm just kidding. Anyways, so this is one of those examples where you could use the words that you know to try to help you to understand something you don't know. Genesis makes us think of something being made, right? And gameto makes us think of gametes. So we're talking about this process of meiosis to make our gametes, okay? So if I didn't do my process of meiosis, what would be the problem? Like, what would be the consequence of that? Well, would my gamete get larger? Well, no, my gamete isn't getting larger. Would the chromosome double with each generation? Well, the point of meiosis was that we were going to split my chromosome number in half, right? I went through 2 rounds of division and made the cells haploid. If the cells were not haploid and they came together, my chromosome number would double each time. So that's kind of where we see the logic. If you need to draw it out, you could, in which you would say, okay, well, I have a 2N and a 2N. So when I fertilize, that makes it 4N. Ooh, that's double what it's supposed to be. So you can, as a student, and you're writing on these questions, you can like mathematically work some of them out, right? Because all you had to know going into this was that meiosis' job is to split the chromosome number in half, okay? The C was talking about chromosome number getting half, so that's basically the reverse of the right answer. And then D was just kind of giving you an extra just fluff answer to see if you even understood what happened during meiosis. So they'll do that oftentimes where they'll give you the complete reverse of the right answer as a way to try to get you to pick it because you don't know the answer, so you're just going to guess. So let's make some good educated guesses. Let's draw some stuff out. Let's make sure that we are good. So free response question. Ooh, yay, free responses. So we're going to do the first part of this question today, and then I'll do the second half of this question when we come back together for the other part of Unit 5 and then all of 6. That'll be in, I think, 2 weeks. Frogs are going to breathe through their skin. And so if we're looking at this plant, right, and the diploid number is 4, so 2N is 4. Flower color is controlled by a single gene in which the green allele is dominant to the purple allele, which we don't really know too much right now yet. The plant height is controlled by a different gene in which the dwarf allele, D, is dominant to the tall allele. Individuals, parental, are crossed, giving us the F1 progeny. Okay, I realize now that you probably didn't have all the genetics background, but we're just going to ride with this. So we construct a diagram to depict the 4 possible products of meiosis that would be produced from the F1 progeny, right? So if we go through the F1, that of course is going to give us, I'm missing something I'm supposed to have in here. We're supposed to have the heterozygotes, right? So if it was homozygous dominant crossed with homozygous recessive, it gave us all heterozygous offspring, okay? So I need to figure out what their gametes were, right? So each one is going to only have 2 chromosomes in it. Why will they only have 2 chromosomes in it? Well, because of the fact that when I went through division, I separated them, right? So I'm coming up with 4 different possibilities for my gametes. 2 of them are going to have a capital G. 2 of them will have a lowercase g, because of course I had capital D and its duplicate, lowercase g and its duplicate, and then I divided to make my 4 cells. So each one will have a capital G, 2 of them will have capital G, 2 of them will have a lowercase g. And then when I think about my dwarf, 2 of them are going to have the capital D and 2 of them will have the lowercase d. And this is all the different combinations you can make with capital D and lowercase g, or you get the idea. So this kind of had a lot of tripping up for students on the AP exam, because I believe they had trouble with their drawings, as well as they had trouble understanding that meiosis was going to make haploid cells. So I wanted to make sure that I went over that. Oh, there's that F1 I was talking about. I must have linked in the wrong spot. Sorry, y'all. So then with mitosis and meiosis, they're forms of cell division that produce our daughter cells containing genetic information from parent cell. Describe 2 events that are common to both mitosis and meiosis that ensure the resulting daughter cells to inherit the appropriate number. So you have to think to yourself, okay, what happens in mitosis and meiosis? There's the same thing that will allow the cells to get the right number of chromosomes. Well, number 1, we go through division, right? We go through replication, right? So that happened during interphase, before we went through this division, right? I have cytokinesis. Cytokinesis takes place after division. They both go through the metaphase. They both go through anaphase. You kind of have to bring all that in together, talking about how your spindle elements will attach to chromosomes, talk about that the chromatin condenses, talk about that in metaphase, your sister chromatids will align on the metaphase plate. It talks about the separation of our chromatids for our daughter cells. We talk about that you have the M checkpoint. Remember, we talked about the M checkpoint's job was to ensure that I had all of my kinetochores, microtubules attached to those sister chromatids. That we go through DNA replication before mitosis and meiosis. Talking about cytokinesis is going to separate my daughter cells, after mitosis and meiosis. So all of these were things you could have put on the prompt. You only have to give 2. Now, when talking about free response questions and they ask for 2, the readers are only going to be able to score your first 2 answers, okay? So when you're writing for response, say, the first similarity is, the second similarity is, so that we as the readers know exactly what you're telling us are your 2, okay? So a lot of times students might write like a beautiful sentence, and in that sentence, they put one wrong thing and one right thing. But both of those could have potentially been scoreable things, and one of them was wrong. So you automatically knocked out your ability to get that second point. So that's why I say you need to write it out. So the first, the second, okay? So then the second question asks us, the genetic composition of daughter cells produced by mitosis differs from that of the daughter cells produced in meiosis. So they're basically saying that they're different. Why are they different? What features happen during mitosis and meiosis that cause them to be different? They're asking you for differences. So you need to mention both mitosis and mention meiosis. You can't just say, well, crossing over happens in meiosis. And you didn't answer the question. So you have to talk about both mitosis and meiosis when you talk about these. So the differences we see, of course, number of divisions, right? So mitosis has 1 division, meiosis has 2 divisions. In mitosis, the cells are going to be, the daughter cells will be diploid. They're genetically the same as the parent, versus in meiosis, they are haploid, and they're going to be genetically distinct from their parents. When we talk about mitosis, the chromatids separate during mitosis, and they will only take place during meiosis too. Crossing over does not happen in mitosis, but it does happen during meiosis, specifically prophase 1. Homologous chromosomes separating does not happen during mitosis, but it does happen in meiosis. That would be anaphase 1. Talking about independent assortment. Independent assortment takes place in meiosis, but not in mitosis, specifically in metaphase 1. So you have to mention both mitosis and meiosis. And again, it asks for 2. So make sure you specifically say the first, the second. So this brings us to our kind of our Q&A part, that if you have any questions, you can ask those questions in the chat and I will answer them. So that I know that we need to kill a little bit of time because, well, you know, there's the lag. I want to go ahead and mention that AP Bio Penguins is a website, right? And on this website, you will find everything you could possibly want to study and more. So I've got all my little content TikToks. I've got quizzes, games. I've got the review sessions from last year's FRQ Fridays where I go over different FRQs. I've got all of my Marco Learning sessions linked, as well as you can access the PDF from my PowerPoint. And I've converted everything, made into like these little, like at the very end of it, there's a one pager. You can print it front and back, and it's got everything from the notes. I've got the daily review. So every day, or Monday through Friday, I post on my Instagram questions. So there are folders that take you to all of those questions. So you can access them even if you haven't started reviewing yet. You can access those now to be able to review. There's all of the live sessions, as well as you can go to the previous year's reviews. There's so many resources here. So I want to just make sure that I pointed that out to you. Also kind of thinking about that you need to make sure that you're following Marco Learning on your Instagram, as well as on TikTok and then YouTube, because that's how you're going to get notified about whenever they have any of these sessions, as well as any of their other sessions. So, you know, AP boot camps are coming up soon. So make sure that you do check out their TikTok and their Instagram to know about their AP boot camps that they're running. I think there's 4 different weekends that they're doing them. So you get like 4 sessions for it. And it's a great resource to help you as you're kind of getting ready for your AP exams. So any questions? As of right now, I don't really see any questions in the chat. I do see an old question that said, would you be able to use Punnett squares in order to get the different combinations? You could use a Punnett square and would be able to make that combination. There's really no reason to go through that because that would have taken some of your time, as well as the question requires you to actually have the circles and fill them in. But yes, you could have made Punnett squares to help your own thought process to kind of get there, but that wouldn't actually be a scoreable response. And so with that, I see that there are no other questions in the chat. So I'm going to say, I hope you all have a wonderful day. Remember, AP Bio Penguins are dressed for success, and I'll see you in 2 weeks.